Okay, hello everybody. Sorry for the slight delay in us getting started. We're just having some technical issues, but I think we're good now. So welcome to the Neo4j Online Meetup. I think we're on number 32 now. So welcome if you've been before, and also welcome if this is your first time. For those of you who this is the first time, today's meetup is going to be presented by Daniel Himmelstein, who's built a tool called Hetionet. Hopefully I pronounced that right.
Hetionet, yeah.
Hetionet. So he's going to be explaining that to us. There's probably going to be some code in the demonstration, so that you can properly see that. You'll want to set your quality to be 720p or higher, which you can do in the bottom right-hand corner of your screen. Other housekeeping. If you've got any questions, obviously we've got the chat on the right-hand side, so you can ask any questions on there. Or you can ask them in the Neo4j Slack channel. With that, I guess I'll let Daniel introduce himself. So maybe you could just give us a quick introduction to yourself and how you got into Neo4j and that sort of stuff.
Certainly. Well, thanks for having me on here, and the fact that we were only 7 minutes late after all the technical difficulties we encountered is a miracle. So glad to be here. So, a researcher. And I started using Neo4j when I was getting my PhD at University of California in San Francisco. Actually, I went to like a local Neo4j meetup. So I guess it's cool that I'm now leading one online. But I work with integrating lots of types of data into networks. And what really stuck out for me was that Neo4j does really well with handling types. So when I started using Neo4j, I didn't even necessarily need a database. I just needed something that would deal with networks where the relationships and nodes had types. And all the database features of Neo4j were actually just a plus at the beginning. So when I go through the meetup today, you'll sort of follow the trajectory I took and the applications that we use Neo4j for. But currently, I'm speaking to you from the University of Pennsylvania, where I'm in the Green Lab, which is a sort of data science and biomedical research group here. So really nice to be here. And I know Neo4j is catching on in academia and research and pharmaceutical research, biology research. So I really hope some people take something out of this and can use something from it.
Cool. Okay. Well, welcome, Daniel. So I guess we should. I guess I'll hand over the presentation rights to you, and you can take us away.
Nice. I have the rights.
I see you've got a link. There's a link on the chat if you want to follow along with Daniel's presentation as well. So anybody can have a look at that.
Cool. Yeah. So I am going to begin the screen share. Hang with me a moment while
cool. I can see it now.
Okay, so
yeah,
now
cool.
You should be able to see this screen. So I put I put the materials for today in a GitHub issue, which I posted in the chat. But if you want to type it in, it's github.com/dhimmel/rephetio, REPHETI.o, and then /issues/3. And you can also post to this if you have any questions. That would be the best way probably to ask questions. So why don't we start? Today we'll be going over something called Project Rephetio, which is a study that was recently published under the more formal title of Systematic Integration of Biomedical Knowledge Prioritizes Drugs for Repurposing. And Neo4j was a large part on the computational front of what we did in this study. And we'll break it down into two parts. But first of all, if at any point you're interested in more details on this study, it's also available on ThinkLab. Following this link, ThinkLab is a site where we had project discussions while the study was ongoing. So you can see all the discussions here, and some of them I'll link to today or go to. And that has the complete detailed information on the study. And you can also read the manuscript at this link using our ManuBot tool, which is a tool for writing papers or documents on GitHub. So while it's not really that related to this meetup, if you're looking for a really technical and hackable way to write scientific papers, check out ManuBot. So the two parts of Project Rephetio are first creating Hetionet, which is a network of biomedical knowledge. And the second was using this network, or Hetionet, to predict new uses for existing compounds. Compounds is synonymous with drugs, essentially, here. So I will go over these two things separately, and we'll start with some background slides on Hetionet. So actually, we'll start very quickly with going back a little bit before I created Hetionet and tell you how I got interested in networks. So here's my Facebook friendship network. All it has in it is nodes, which are users, and connections if the users are friends. So every sort of connection here is a mutual friendship. And knowing nothing more besides this network or graph structure, I was able to cluster it and do an automated community detection algorithm, which was able to really decipher my past social history. So for example, you can see my summer camp over here, which had a lot of people from my hometown, but was separated. You can see when I went to college and how the debate team I was on was a little bit socially isolated, should we say, from the rest of the college. You can see where I went to grad school. My mom has a really big family, which is over here. And what was surprising was under hometown into two communities, and that perplexed me for a little bit until I realized that one community was everyone above my brother's age, and then one community was my brother's age, essentially. I have a younger brother, and I guess he's a social hub, let's say. Anyways, I got more interested in how we could make richer networks, networks that have type. So here's a really simple example of a network that has type. Obviously, graphs are composed of nodes and relationships, and this is a network of the University of Pennsylvania and the courses and how I've interacted with them. So, for example, nodes and relationships can have type. Here in green, we have a person node, which is Daniel. We have two course nodes, Epidemiology 600 and Computer Information Systems 550. And we can have relationship types. So for example, I lectured in this course, and this course is at the institution of University of Pennsylvania. So anyone who uses Neo4j probably very naturally thinks about types on networks like this. But actually in biomedicine, the most common way people do things is with graphs with no types. And that's because the algorithms are more advanced for those homogeneous networks. Although when we want to represent something rich, you know, it's important to have that heterogeneity. So as you know, you can have properties on the nodes. You can have other types of nodes like locations, and you can even have multiple types of relationships between the same types of nodes. So for example, you could have a professor of a course or a guest lecturer, or even students. So now how does this apply to biology? We wanted to teach the computer biology. We wanted a way of encoding biological knowledge from the past, essentially 50 years of study, into a single data structure that we could operate on and extract information or make predictions on. So we created Hetionet. This is a metagraph of Hetionet, which is also called a data model or schema, and it's showing you the types of nodes and types of relationships. So note here how we have compounds and we have diseases. And we have this relationship for when a compound treats a disease. And that's actually what we're most interested in for the Project Rephetio study. But the network also has a lot of other pieces of information. So here, this indicates whether a compound binds to the protein of a certain gene. But there's also other relationships between a compound and a gene, such as the compound upregulates a gene, meaning that when a patient or a cell has the compound, more of that gene is transcribed or expressed. So for people who don't really know biology, this may be a little bit intimidating, but don't worry. Just think about this as a network with many different types of biomedical entities and connections between them. It's also cool that we were able to connect information across many different scales. So for example, genes are molecular. They exist inside a cell or at the cellular molecular level. Whereas here we have the side effects caused by different compounds or drugs. And that's really something that's experienced at the organismal or tissue or organ level. Same with the symptoms of a disease. So what these heterogeneous networks allow you to do is really connect information on a lot of different scales and put a lot of diverse types of knowledge together. We call it a hetnet, short for heterogeneous network. Whenever you have a network where the nodes and relationships have types and more than one type. Here's a bird's eye view. Each node here is a tiny dot, which is hard to see, but if you looked at all the genes, of which there are 20,000 protein coding ones in human, they'd all be a little dot. And then the lines here represent the relationships. So we created Hetionet version 1.0 as a hetnet of biology for drug repurposing with 50,000 nodes of 11 types. If you go to the previous slide, you can see the 11 different types and 2.25 million relationships of 24 types. We should note here that in Neo4j, you can have multiple labels or types per node, but this network only has one type or label per node. And this integrated 29 public resources, which themselves had combined knowledge from millions of studies. So we took essentially the knowledge from tons of papers, databases that a lot of people worked very hard to create and put it all together. And there were some challenges. If you're creating networks, you'll probably face these. We did it in Python using Jupyter notebooks and GitHub and pandas, really. But what's important is that when you're creating nodes, that you use standardized vocabularies. You don't want one piece of relationship you use to have diseases in one terminology and another one in a different terminology. In that case, you'd have multiple, essentially, nodes for the same disease. And that's a big problem come query time. So you got to do that hard work when you're making the network to use a standardized vocabulary, map everything to it, and apply stable, unambiguous identifiers. So for our relationships, we wanted omics scale. So we used a lot of literature mining or high throughput experimental technologies, and always try to avoid manually mapping things. If you manually have to do a step, it's probably not scalable. And we use version data dependencies with GitHub URLs that use a commit hash. This is a bit technical, but so at the beginning, actually, we weren't using Neo4j. We were using a package that I made called Hetty, which at that time had 5 stars on GitHub and 2 forks, and then we discovered Neo4j with 3,000 stars and 1,000 forks, and we started using it. So we actually still use the Hetty package, which is a Python package, but more as a wrapper around our Neo4j usage. So the main database is now in Neo4j. And We created a public Neo4j instance, which we'll be referencing throughout this meetup at neo4j.het.io. And well, I guess I'll go over the cool points of this right now because we're here. It uses a customized Docker image, so it's really easy to set up on whatever cloud host you want. We use DigitalOcean with SSL for Let Encrypt. And what was really crucial is that Neo4j has a read-only mode with a query execution timeout. So we essentially provide this Neo4j database free to the world, and it hasn't had too much trouble thus far. I hope it stands up to everyone in this meetup. But people thought we were kind of crazy when we said we want to make a public authentication list server. But it's working good so far, and I actually think the Neo4j browser does really well as just a public front end to a single database. So if you want to do this for your data or your database, you can get some information here. So now let's get to the specifics. Okay, so we will be going to that Neo4j browser I mentioned. The URL is neo4j.het.io. And what you'll see here is the traditional guide that you're accustomed to when starting a Neo4j browser. You'll also see how we've changed this configuration setting, autocomplete to false. That just means it doesn't fill in relationships between nodes unless you specifically query that. You can change that under the settings here if you want to change that back. And here is actually a custom guide that we wrote. It's cool that Neo4j allows custom guides. So what this does is it shows the metagraph, which we showed you before, but it also has many different panels. So for example, if you wanted to know how many of each node type there are in the network, you could run this query. And there you go. You can see we have 21,000 genes, 11,000 biological processes. You could do the same thing to count the relationships. So the most common relationship is when a gene participates in a biological process, of which there are almost 600,000 of those. And what's cool is the random relationship query. Now, this takes a random relationship for each type of relationship and returns one. So it's a good way to see, what are the relationships in the network. It's more computationally intensive because it actually has to go through each relationship.
Daniel, we've got a question for you from the chat.
Cool. Do you want to say it or the?
Yeah, we can do this one now. Then there's another one for the end. So this one is your standardized vocab public? Is the question.
Yes, I should mention it's all public. Nothing in this project is private. And we only used resources that we felt that we could make public and that were public to begin with. So that was very important for us because we wanted to create a resource that other people can reuse. And actually, we did deal with a lot of licensing issues because a lot of people in academia didn't realize that data may have copyright and stuff. So we tried to the best of our ability to really make sure that we could redistribute what's in the network. And the vocabularies, yes, are all public. And as is this network, you can actually access it on GitHub
is there a password
in different
on
formats.
That? Is there a password on that?
No, there is no password.
Okay,
so everyone should be able to see this.
Okay, cool. And then I've got another question, which is, have you considered epigenetic phenomena? Which I'm not sure whether that's something.
No, that's a good question. We don't have too much epigenetic information in there now, but you could add it. You really just need a resource that is essentially omics scale or systematic. We would like to put these single nucleotide polymorphisms in the network in the future, and that would have a lot of opportunity for epigenetic style relationships. We don't have that yet. Back to a previous question, the network's on GitHub in different formats. So this is dhimmel/Hetionet. We have it in a JSON format, a Neo4j format, and just a tabular format. So under the Neo4j, you can see a compressed version of the database. And then here's the Docker information for essentially rerunning this server locally. So what we have here is we have the random relationship of each type. Sorry, Mark, did we have any more questions before I jump back in?
No, you covered them all so far.
Cool. So what do we have here? And this is not rehearsed because these are random. So liver cancer presents this symptom, which is, you can see down here, ecchymosis. I don't happen to know what that means, but maybe someone listening does. What do we have here? We have this drug, which is pentamidine, causes a side effect of tension. Well, here we have two drugs. This is that desogestrel is chemically similar or resembles this other drug, which is gestadine. And you can actually see the chemical structure as a property here. That's what the INCHI refers to. And that's what we use to calculate that resemblance. Here's a, let's see some other interesting ones. We have a disease here, osteosclerosis, and that localizes in the anatomy or tissue of tensor tympani. Essentially, we're through text mining, we've inferred that this disease affects that tissue. So yeah, I encourage you to play with this a little bit more to get a feel for what is in the network. So now let's go back to this guide here. I'm going to go to the second to last slide. It's a good idea to load the custom style, which will make the nodes the right colors so you see the same things that we're seeing here. I've already loaded it, but if you haven't, you can load it in that way. So now we'll go over some really basic Cipher queries, and we will get to the more advanced Cipher queries later. So here what we're going to do is we're going to search for the disease called lung cancer. It's just going to return a single node, so not too difficult of a query. In the Neo4j browser, you can double-click and expand the relationships. It probably didn't show everything because there's too many. I think it probably cut it off at 100. But you get the idea. That query is equivalent to this query. In general, I like to separate the distinct where statement because I think it reads a little bit better. So here we can do something a little bit more advanced where we find all the tissues essentially that lung cancer affects. So there's lung cancer in the middle, and it's affecting the epithelium, the bone marrow, the bronchus, the lung. This query, oh, no, we already did that one. We can find all the genes associated with spinal cancer. So we're saying the disease should be spinal cancer, and the relationship type should be associates, and to return that. So we found 7 genes, and what's cool here is you can hover over the edges to see a little bit of metadata. So this one's actually a little bit boring. It's all from the diseases database, but oftentimes you'll get databases where you have more metadata. Let's go here and see some more association edges. Okay, here we have a little more advanced of a query. So we are going to look for when the source disease, which we're specifying as liver cancer, is associated with a gene that's associated with kidney cancer. Essentially, we're finding genes that are associated with both liver and kidney cancer. And then we are returning the symbol and the name and a URL, which you can go to learn more about the gene. So let's try running that. And there you get the table. So it found 36 genes that are associated with both, and here they are. So in this example, it's similar to what we did above. We're interested in the mitotic spindle checkpoint, which is a biological process, and we're interested in the genes that are participating in that. Now, for something a little bit more complex, we're going to find the same query where we're looking for genes that are participate in the mitotic spindle checkpoint process. And we also want to limit those to genes which are expressed in the lung. Potentially, we could be interested in the mitotic spindle checkpoint in the context of lung cancer. So we'd want to make sure the genes were expressed in the lung. And so here you have these genes that participate in the biological process that are also expressed in the lung. Cool. So now for the more advanced stuff.
Another question for you, Daniel, is
do
you know of any researchers using the tool you've just been showing?
Yeah, so I think a lot of people have started using it in addition to the research we've done on it. We can see the logs, but I think it's had it gets maybe 1,000. It gets thousands of views per 2 months, maybe.
Okay.
So people are using it, and actually we recently started logging the queries people do, so that'll be something cool to look at to see kind of what people are using it for. But yeah, we do have people using it. So there's some more advanced queries here on this discussion. I like this one, so let's. The one I'm going to show you I like because I think it shows you how powerful Cipher is and how powerful hetnets or networks are generally for doing traversal type queries. So this is something that I think would take, a lot of lines in something like SQL and would be quite miserable. So we are looking for the disease multiple sclerosis, and we're interested in when that associates with genes that interact with other genes that participate in the retina layer formation biological process. And not only that, this edge here, this association edge, we only want those edges that are from a type of study called GWAS. So we're saying the source of this edge has to be in part the GWAS catalog. And not only that, we want to make sure that the disease here is affecting a tissue that upregulates this gene, which is node 2. So we're saying we're only interested in genes that are upregulated in the tissues affected by multiple sclerosis. And we want to return the paths. So there you go. Very quickly, we have found these paths which satisfy this, these bespoke constraints. Okay, so we've gone over the cloud hosting and we went over the GitHub. So now I'm going to transition to Project Rephetio.
I do one question just before you start that bit. So The question is, you mentioned that some of the relationships in the graph are derived from doing text mining. And so the question is, how did you do the text mining? What method did you use?
Yeah, so sometimes other people did the text mining, and we just sort of took the results. But the main one that we did ourselves was actually co-occurrence. So looking at different papers and to see whether two terms co-occurred more than you'd expect by chance. So for example, is the symptom fatigue seen more with the disease MS, multiple sclerosis, than you'd expect by chance, given the sort of independent probabilities of those of that symptom and that disease. And while that's not the most sophisticated method, we found it worked pretty well. Currently, we have projects to do more advanced text mining. If you go to the repository greenlab/snorkeling, you can find the state of our current research. But in the past, we did co-occurrence and found that it worked well. Oops. So Project Rephetio, this was how we applied Hetionet to make drug repurposing predictions, or predicting new uses for existing drugs. This is really an area of interest because if a drug's already approved, you know that it's safe, and it's a lot quicker and cheaper to get it approved or used for a new condition rather than making a new drug from scratch. And it turns out that a lot of these drugs do a lot. They call them promiscuous in terms of the targets they have. And because of that, oftentimes we only use them for one or two diseases, but they potentially could have a lot more effects. And a lot of drugs, we've sort of observed that they work through serendipity or just chance. So could we take a more systematic computational approach to determining when a drug will treat a disease? So Hetionet contains 755 treatments. These are known compound instances when a compound treats a disease. And that's obviously a lot smaller than all possible compound disease pairs, which is 210,000 in Hetionet. So we did systematic drug repurposing, where we wanted to compare the therapeutic utility of different data types, identify the mechanisms of drug efficacy, and predict the probability of treatment for all the compound disease pairs. So the way we did this from a machine learning approach, which is not a big part of this talk, but it'll help you understand why we're using Cipher the way we're using it, is that we looked at certain types of paths. So each column here is a type of path. For this one here is compound binds a gene associates with the disease. We call it a metapath. And for each metapath, we looked at, or for each compound disease pair, which are the rows here, we looked at how common that type of path was, how many paths of that type there were between the specific compound and disease. So for example, with midazolam and epilepsy, this path is quite common. That's why it's in a darker color. And we end up fitting a model where we know that some are known treatments and the other ones are non-treatments. So we try to see what types of paths occur more frequently between the treatments and the non-treatments. And we were able to predict relatively well using this method. So for example, we were even able to predict applications or treatments that were currently in clinical trial. And so for one example of a single prediction, which we'll go over today, bupropion is a medication that was first approved for depression in 1985, but in 1997, it was approved for smoking cessation. So the question is, can we predict this repurposing from Hetionet? And it actually was total luck that people realized that the patients who were depressed and taking bupropion for depression happened to want to smoke less. in. So could we predict this? And we actually did give that a high prediction in the 99.5th percentile for nicotine dependence. And what our approach does is it actually picks out the paths that are behind that prediction. So one is that bupropion causes side effect of terminal insomnia, which doesn't mean that it kills you. It just means that the insomnia, you can fall asleep fine, but then you wake up in the middle of the night and can't go back to bed, which is also caused by varenicline, the only other FDA-approved treatment for nicotine dependence. They both bind to this CHRNA3 gene, and this gene happens to have been found in a genetic association study to be associated with nicotine dependence. And in addition, this gene participates in a lot of pathways where other nicotine or smoking associated genes participate in. So these are the top 10 paths that our approach identifies as why we think bupropion will treat nicotine dependence.
Got another couple of questions for you.
Yeah.
So the first one is, what sources were used to establish the paths?
Can you?
So the question is, what sources were used to establish paths is the question.
So a path is nothing more than a sort of chain of edges. So potentially they're asking in this example that I showed, what databases were behind all the edges. We integrated 29 different databases. So I can't go over them all now, but something like the compound binding to the protein of this gene was from databases such as BindingDB, DrugBank, Drug Central. Those were the 3 databases there. This side effect compound relationship was from Cipher. I don't want to spend much time on that because that information is available.
Yeah, I think the person's happy with your answer. So we're good with that.
Cool.
And then the second question was, have you tried running any graph algorithms over this data set? For example, would there be any community detection or clustering or that type of thing? I think you mentioned
we haven't done that yet too much because those algorithms don't often scale to the hetnets or networks with types. If you just run like a regular community detection, it's not going to appreciate the differences between the different relationship types. So for example, different relationship types have totally different degree distributions. Some are more prevalent than other ones. And if you don't take that into account, you'll get a junk result. So not that those aren't very interesting applications, but
so what would you have to do if you wanted to use one? Did you have to do it just on a specific set of relationship types inside the graph?
Maybe, but that's not. Yeah, you probably, if you wanted to use one of those existing algorithms, there are researchers who are working on extending these algorithms. And I should note that what we did is extend a relationship prediction algorithm. Okay. So yeah, we do have an algorithm that we have that works on the hetnets, but it's specifically for predicting missing edges. And that makes sense for us because we're predicting essentially missing treatments.
Cool.
So yeah, so here's our browser, het.io/repurpose. And so for example, we could search for epilepsy and we could see all the predictions. And why don't we just stick with bupropion, because that's what I mentioned before to you. So we can see it here. Here's the prediction, and we could go in a new browser. If we click that link, we can actually play a guide just for this prediction. So if you click on this command, it'll show you those 10 paths. Well, I'm not sure why something looks to be different than the visualization I showed you before. I'll follow up with that, but I was expecting to see those side effects in here. Not sure why they didn't show up, but I will investigate that later. You can go here and see the different contributions by paths and the different contributions by metapath. So I'm not going to go over this graph just here, but it's going to break down what the algorithm we use in a more simple way. And it actually was made during the last Star Wars release, The Force Awakens, so it's Star Wars themed. And I guess it's pretty appropriate that The Last Jedi will be coming out soon. This actually won the open data category for the competition that year. Okay, so now let's go over the cipher that we use. So we're going to focus on just quantifying the relationship between bupropion and nicotine dependence, specifically along a certain metapath or a certain type of path, which is that a compound binds to a gene that participates in a pathway that that the disease participates in. The disease here being nicotine dependence and the compound being bupropion. So let's take this query. Okay, so I know this is a little bit unmanageable, but what we have done here is we have found all the paths of that type. So let's go over the cipher. You have the metapath definition, that it's a compound which binds to a gene. Note that we don't actually have to put gene here, so we could go like that. But we don't have to, because we made sure that each relationship type in the network is unique. And we did that by appending the source and target node types. So for example, here you can see there's several types of relationships that have participates, but they're disambiguated with this little suffix here. And that makes the queries more efficient, because if you were to have gene here, there would have to be a filtering step in order to check whether this node is a gene. But we can sort of encode that right in that relationship type. So that's a little trick. So we have the path here, and we're just saying that node 0 has to be bupropion and node 4 has to be nicotine dependence. And then below we returned all the paths, but we could also, if you just want to know the path count, we could do this. So there's 142 paths of this type. And actually, what I just said is potentially incorrect. There are 142 trails. I'm not sure if anyone, this is like mathematicians use this terminology. A path technically doesn't have duplicate nodes, whereas a trail can have duplicate nodes. And if anyone knows what Neo4j does by default, is when you're matching a pattern, it allows duplicate nodes. It does not allow duplicate relationships. If a sort of traversal has duplicate relationships, it's called a walk. So Neo4j doesn't find walks, but by default will find trails. We're interested in paths, so what we end up doing is we know that there are only 2 nodes of the same type here, which are the 2 genes. One of those genes is node 1. So if we say we're node 1, does not equal node 3, we can actually get paths instead of trails. And in this case, there were no trails with duplicate nodes, essentially. So the path count is the same as the trail count. Let's just show that to you in a table format. So what we will do is use this node function, which extracts the nodes along each path, and then we'll just take the name. So here you have your 142 rows with the node names along the path. And you don't need to know the relationship types because those are all the same in this query. One other thing we found helpful was specifying the join index. So I'm going to go back to this query. And what do I mean by the join index? Well, here's a trick. You write profile here. This should work. So what is this saying? This is giving us a query plan. What it's doing first is it's looking up the compound. Then it's going from the compound and it's finding all of the binds relationships starting from that compound. And then it's going further out to the participates. It's essentially going along the path in a breadth first search and finding all the paths. So what you can do is actually start the search from both ends and then join the results in the middle. So this will maybe it has to go here. There we go. So now you see this cool split structure. Here you're starting the query from the compound. Here you're starting it from the disease. And then you are in the middle, merging all the sort of subpaths that you found on the common node. So this oftentimes is a lot more efficient. So we can actually see the overall efficiency. This query, when you specified the join index, took 392 database hits, and this one took 123,000. So Neo4j will often pick one by default, but we found it was often best to specify it, given that a lot of these queries were computationally intensive. And a cool idea for the future is I think it may actually be possible to determine the join index during runtime. So you can see this GitHub issue for that more radical proposal. So the next thing we're going to do is extract degrees along each path to compute what's called a path weight. And the purpose of what we'll be doing here is that some paths go through very high degree nodes, and that's oftentimes not very informative, because remember that a high degree node could be connected to everything in the network, and therefore every path would go through it. So if a path is going through a high node, we want to sort of give it less weight. So what we do with this kind of cool cipher here is for each node, we calculate the degree. So this is node 0, and we know the relationship type is binds. So we say how many nodes, or how many relationships of that type do you have? That's what size does. A quick note, if you were to go, if you were to say the type here, that this has to be a gene, which is unnecessary because this encodes that information. This would be like thousands of times slower because right now all it has to do is count the degree, whereas here it would have to filter based on the gene. So there's a lot of tricks with Cipher to get things computationally efficient. So we go through each sort of endpoint of each relationship, and we calculate the degree. And what that gets us is it gets us, for each path, something called degrees. So actually, I think it'll be helpful to do this. Okay, so this shows you here, for each path, we have the degrees along the path. So that means essentially bupropion is binding to 12 genes, whereas CHRNA3 is binds or is targeted by 10 drugs. So for each relationship, you have two sort of degrees. And then this pathway highly calcium, blah, 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 or essentially this gene participates in 10 pathways, whereas this pathway contains or has 11 participating genes. So now we can, now that you get that part, we can do this where we're multiplying all these, we're putting those degrees to a negative power and multiplying them together. So what that does is paths that go through higher degree nodes get a lower weight. I think we actually, well, so this shows you the top 10 paths based on their weight, but I think I like showing the table. So
Hey, so I guess we should probably think about wrapping up soon. We're close to
it's
the end
one.
of the,
okay.
Yeah, but I got a few questions maybe we can wrap up with.
Okay, cool. So actually, I do think we got through all the meat here. Let me just show one last query, and then we'll get to your questions.
Okay,
This
cool.
will be quick.
Yeah.
So here's, in the end, we can run this one query, which finds those 142 paths, and then finds this degree-weighted path count, which has taken into account the degrees along each path to downweight the paths through high degree nodes. And so this is the basic sort of feature extraction we did to power Project Rephetio. So now let's get to those questions.
Cool. So the first one is, do you have statistics of results of actual repurposing and how it matches predictions made? What is the success rate, treatment producing desired effects percentage?
Yes, hold on. I'm going to come back so we can see each other. Okay, you can see me now. So,
oh, cool. Yeah.
Yeah, we found it was highly effective at predicting known treatments and also effective at predicting the treatments in clinical trials. We also did a little analysis with epilepsy. Epilepsy is a cool disease because it's pretty easy to test whether a drug will, what effect it'll have on epilepsy. There's good animal models. And we found that like of our top 100 predictions, about, I think 80 of them were actually effective for epilepsy. So I think it was very promising. And we hope that people follow up experimentally on our predictions. one difficult thing is a lot of the predictions of pharmacologists could probably say, yeah, I agree with that, and maybe I could tell you that myself. So we still need to get to a point where it's integrating this really, or drawing more on this really high throughput data that a pharmacologist could never conceptualize because it's millions of data points. Now we've essentially done a really good job of being an automated pharmacologist to predict 210,000 potential treatments. But it's still something that probably a pharmacologist could do if you gave them infinite time.
I suppose it gets interesting though, doesn't it? If you do ones which are really difficult for them to work out, how do you know, like you don't have anything to compare it to say?
No, but because you can see those paths behind it, someone can look, is that reasonable?
Okay.
You know, some of them, we found a pattern, and while that pattern generally holds, like a pharmacologist maybe will say, in this case, the pattern doesn't really make sense to be applied. You know, that type of that specific path it, is targeting a gene which is actually just involved in processing the drug and not actually in drug efficacy. So it is good to manually look at why the predictions are being made.
Cool. All right, I think we've answered the question. So next one is, so first of all, the person says, thanks for the presentation and open sourcing the code. So they've asked a few questions actually as we've gone on. And next question is, how often is Hetionet updated?
Yeah, so it's not continuously updated. And that's something that we'd love to move toward. Although it's very hard because like we rely on all these databases and they change your formats. So there's going to need to be a lot more standardization and rigor with bioinformatic databases before this can be entirely automated.
Yeah.
Currently, the next probably major update will not be for a bit of time, but should include a lot more of the text mining. And it should allow you to go from a relationship back to the actual sentences in papers that we think support that relationship. There are other groups working on actually updating Hetionet. So that if you have a specific need for a certain part of the database to be updated, you could always do yourself. Did I freeze?
Yeah, I think so. Although I can still hear you, so I guess that's good. Last question, if you're still there.
Yeah, I am there.
Okay, cool.
Hear me.
Have you considered using NBXBio as a source database?
NBXBio. Let me Google that quickly.
NDXBio. I'm not sure if I'm saying that correctly.
Okay. Yeah, I'm still Googling because I don't want to give the wrong answer. I believe that's a project that is sort of like a platform for storing networks, if I'm not mistaken. Produced probably in California, also if I'm not mistaken, maybe not. I looked at it a little while in the past, and it wasn't clear to me that it could handle the size of the network, but if it can now, and it can reduce some of the burden that we face integrating all the data, that would be great.
Yeah.
It's more than a one-person job to create all of these networks and to integrate the data. So yeah, resources like that can make these into community projects and bring individual efforts all into one central place. Sort of like how open source software brings a lot of commits together. That'd be cool.
Cool. All right. Well, I guess we don't have any other questions for the moment. So I'll give people a few seconds. I think it's a bit of a delay. So if you enjoyed the presentation, don't forget to like it on YouTube so that more people will see it. And I'll just give you everyone a few seconds to ask some more questions. And during that rambling by me, we have a new question, which is, have you considered the aspect of nutraceuticals to integrate within Hetionet, as in comparisons to current drug compounds, for example?
I don't know what a nutraceutical is.
Okay, maybe we can. I mean, we can always do some of these. Maybe we can do some of these questions either on the GitHub issues or the.
Could be So I think a nutraceutical is referring to essentially a compound that is not synthetic and therefore not subject to FDA regulation. And if I understand that correctly, that the resource we use, Drug Bank, will include compounds like that. And there should be some in there. Something like caffeine or something would be in there.
Okay.
But yeah, I'm not sure that's an exhaustive answer, but I guess you could look through the compounds at het.io/repurpose. And there's about, I believe, 13 or 1,500 of them. And it is, you know, I would like more. There's obviously a lot more chemicals or compounds. But we really need things that are highly connected and that have enough public information on them. Since we're dealing with networks, if something's not connected, it's there's no meaning to it in a network sense. Cool.
All right. I think we can probably call it a day then. So thanks to everyone for joining us to watch. And again, thanks to Daniel for taking out the time from your day to present. I think it was really interesting.
Thanks. I'm really happy that we got everything working. And sounds like from the questions, there was great participation. And if you have any more questions, the best place is a GitHub issue on the relevant repository. We happily answer them all. So thanks.
Awesome. Enjoy the rest of your day, everyone. Bye.
Cool. See ya.